9th standard students, in science, I am going to continue the lesson Organization of Tissues. In this video, we are going to see about cell division. What is cell? Cell is the structural and functional basic unit of life. Every living organism is made up of cells. All cells are produced from other cells by the process of cell division. Cell division occurs when one cell divides to produce two new cells. Unicellular organisms use cell division to reproduce. Multicellular organisms use cell division for growth and repair of damage such as wounds. What is cell division? Cell division is the process by which a parent cell divides into two or more daughter cells. Cell division has three main functions. One is the reproduction of an entire unicellular organisms like bacteria, yeast. Then the growth and repair of tissues from damages in multicellular animals. And the formation of gametes. Gametes are eggs and sperm for sexual reproduction in multicellular animals. There are three types of cell division: amitosis, mitosis, and meiosis. Amitosis is the direct cell division in which the nucleus and cytoplasm divide by constriction without the formation of chromosomes. Mitosis is the process of making new body cells. So mitosis is the indirect division. Meiosis is the reduction division, which creates egg and sperm cells. Now let us know about types of cell division in detail. Amitosis. Amitosis is an simplest cell division. Amitosis is mostly observed in unicellular organisms like bacteria, yeast, amoeba. During amitosis, the nucleus elongates first. Then the nucleus divides by the appearance of a furrow. This furrow is known as constriction. This constriction deepens to divide the cell into two individual cells. Finally, cytoplasm divides, resulting in the formation of two daughter cells. Mitosis. Mitosis was first discovered by Fleming in 1879. Mitosis is a process in which a single parent cell divides into two identical daughter cells. Two identical daughter cells genetically identical to the original cell. All the daughter cells having the same amount of DNA, same number of chromosomes and genes. So, mitosis is also known as equational division. Mitosis consists of two events. They are karyokinesis and cytokinesis. Karyo means nucleus. In this, nuclear division takes place. Cytokinesis means division of cytoplasm takes place. Interphase. Interphase is the resting stage of the nucleus. During interphase, the cell undergoes normal growth processes and also preparing for next cell division. Karyokinesis is the first portion of the mitotic phase, in which division of the cell nucleus takes place. Karyokinesis is divided into a series of phases. They are prophase, metaphase, anaphase, and telophase. First, we will see about prophase. Pro means first. So, the first and longest phase of mitosis is prophase. During prophase, chromatin condenses into chromosomes. And the nuclear envelope is the membrane surrounding the nucleus. This nuclear envelope breaks down. In animal cells, the centrioles present only in the animal cells. The centrioles near the nucleus begin to separate and move to opposite poles of the cell. Next stage is metaphase. Meta means after. So in this stage, chromosomes align in the equator of the cell. The chromosomes line up along the metaphase plate. The metaphase plate is an imaginary line that runs across the cell. During metaphase, spindle fibers attached to the centromere of each pair of sister chromatids. The next stage is anaphase. Ana means up or back. It is the stage of mitosis after the process of metaphase. When replicated chromosomes are split and the newly copied chromosomes are moved to opposite poles of the cells. Telophase. Telophase is technically the final stage of mitosis. Its name is derived from the Latin word telos, which means end. During this phase, the sister chromatids reach opposite poles. The small nuclear vesicles in the cell begin to reform around the group of chromosomes at each end. As the nuclear envelope reforms by associating with the chromosomes, two nuclei are created in the one cell. Cytokinesis is the process in which the cell actually divides into two. With the two nuclei already at opposite poles of the cell cytoplasm separates and the cell pinches in the middle. Ultimately, it leads to cleavage. Then, cytoplasm divides into two. Significance of mitosis. Mitosis results in the production of diploid daughter cells with identical genetic material. Cell divides by mitosis to restore the nucleocytoplasmic ratio. Mitosis helps in cell repair. Mitotic division results in a continuous growth of organisms and organ development throughout their life. Meiosis. Meiosis is a special type of cell division of germ cells in sexually reproducing organisms. It is used to produce the gametes such as sperm or egg cells. It involves 2 rounds of division that results in 4 cells with only one copy of each paternal and maternal chromosomes. Paternal chromosomes from father. Maternal chromosomes means chromosomes from mother. The term meiosis was coined by Farmer in 1905. Meiosis is called reduction division because the number of chromosomes in the daughter cells are half that of the parent cell. Meiosis division reduces the number of chromosomes from 46 chromosomes to 23 chromosomes. Chromosome number is reduced to haploid n from diploid 2n. Here, N describes a single chromosome set. The two sperm cells split again, producing 4 sperm cells, each with 23 or N chromosomes. Meiosis consists of two divisions. They are heterotypic division, homotypic division. Heterotypic division, otherwise called as first meiotic division. Homotypic division is known as second meiotic division. Meiosis 1. In meiosis 1, the actual chromosome number is reduced to half. Therefore, meiosis 1 is also known as reductional division or heterotypic division. It results in the formation of two haploid cells from one diploid cell. Meiosis 1 consists of 5 stages. They are prophase 1, metaphase 1, anaphase 1, telophase 1 and cytokinesis. Prophase 1. It is the longest phase of meiosis. There are 5 different stages in prophase 1. They are leptotene, cycotene, pachytene, diplotene and diakinesis. First, leptotene. In leptotene stage, chromosomes become distinct, long and uncoiled. They occur in diploid number. They contain a series of bead-like chromosomes. During this stage, the chromosomes attach themselves by their ends. These ends are known as telomeres. They attach their ends to the inner membrane of the nuclear envelope. This forms a bouquet stage. Cycotene. In cycotene stage, two homologous chromosomes approach each other and begin to pair. Homologous chromosomes are two pieces of DNA within a diploid organism which carry the same genes. This pairing is called synapsis. Pachytene. During pachytene, the third substage, the chromosomes continue to condense. Crossing over takes place at this stage. At each point of crossing over, a chiasma is formed between non-sister chromatids. Chiasmata is the X-shaped structure formed due to the point of contact between paired chromatids during meiosis. So chiasma is the point representing the crossover where the homologous chromosomes are joined to each other. Diplotene. In this stage, the homologous chromosomes repel each other. The two homologous chromosomes are separated from each other. The chiasmata begins to move along the length of the chromosomes from the centromere towards the end. The displacement of chiasmata is termed as terminalization. Diakinesis. In diakinesis stage, chromosomes achieve their maximum thickness. The chiasmata and nucleolus disappear. The nuclear membrane degenerates and the spindle fibers make their appearance in the cytoplasm. Metaphase 1. The chromosomes move towards the equator. Then chromosomes locate themselves on the equator. In metaphase 1, the two chromatids of each chromosome do not separate. The centromere does not divide in this metaphase 1 stage. Anaphase 1. The centromere do not divide at this stage, but the two poles of homologous chromosomes move towards the opposite poles. This stage is called as dyad. Telophase 1. After reaching their respective poles, the haploid number of chromosomes becomes uncoiled and elongated. The nuclear membrane and the nucleolus reappear. Thus, two daughter nuclei are formed. Cytokinesis is the cytoplasmic division. So, cytoplasmic division occurs and two haploid cells are formed. Meiosis 2. During meiosis 2, the sister chromatids within the two daughter cells separate. They form four new haploid gametes. Meiosis is also called equational or homotypical division because the number of chromosomes in the daughter cells are similar to parent cells. So they are known as homotypical division. It consists of 5 stages: prophase 2, metaphase 2, anaphase 2, telophase 2 and cytokinesis. In prophase 2, the centriole divides into two. Nuclear envelope and nucleolus breaks down and then the spindle fibers appears. In metaphase 2, the chromosomes line up individually along the metaphase plate. In anaphase 2, the sister chromatids separate and are pulled towards opposite poles of the cell. In telophase 2, nuclear membranes form around each set of chromosomes and the chromosomes decondense. Cytokinesis. In cytokinesis, a nuclear envelope forms around each set of chromosomes and then cytokinesis occurs. Cytokinesis producing 4 daughter cells, each daughter cell with a haploid set of chromosomes. Significance of meiosis. In different species, the constant number of chromosomes maintained by meiosis. Due to crossing over, genetic variations produced. Variations means different characters which are transmitted from one generation to next generation. So dear students, I hope you understood the cell division and its types. Thank you students.